All right. So my name is Corey. I am from EBIC, and today I just wanted to give a brief introduction into or about EBIC. I also wanted to give an update on some of our software and computing changes that have been made over the last year, as well as giving a brief update on two of our in-house research areas and how we're hoping to bring lessons learned from these technologies to the user program and share it with the community in the UK. So, as most of you know, EBIC is the UK National Centre for Cryo-EM. It's funded by Wellcome Trust, MRC, and the BBSRC. It's a free at the point of access facility to state-of-the-art cryo-EM. We allocate time based on a peer review application process, and our goal is to run like a beamline using a 24/7 operation model run by expert staffs. So we also have a cutting-edge in-house research program under the leadership of Peijun Zhang. And we also strive to foster development with integrated structural biology in the UK through CCPEM, EMDB, and iNext. So part of this is also providing training to the community. So we've done several training courses, such as advanced data collection for high-resolution cryo-EM. We've also done 2 iNext and FEI-sponsored sample preparation courses, one in January 2017 and one in October of last year. Both were oversubscribed by tenfold, which really shows the demand for this technology in our field. And the last thing is we've started doing BAGS super user accreditation, and I'll talk a little bit more about BAGS later, but essentially we're now allowing users who have passed accreditation to actually collect their own data, and it allows a more flexible data collection for our users. So at EBIC, we have 6 microscopes. We have 4 Titan Cryoses, all equipped with Voltaphase plates, along with Falcon 3 and K2 detectors. We also have a Talos Arctica, which is used mostly for in-house research, as well as development of new technologies, or new methods for us, such as electron diffraction, which I'm going to be talking about a little bit later. And finally, we have the SIOS, which is our CryoFib SEM. So user access modes for the Cryos. So we have 2 routes of access. One is rapid applications, and these are 48-hour blocks of time. They're peer-reviewed every 3 months. Usually the top 30 to 50% of the applicants are selected based on a peer review panel, and we look for about a 3-month turnaround time, and we hope to bring this to as quick as 6 weeks. The next route is block allocation groups, or BAG access. And what this does is it allows us to give multiple sessions to research group consortia, where we assign time every 6 months on a rolling, like a 2-year time period. And what it allows you to do is that every month you get a certain amount of time, and then within your group you can split up that time, and it allows for more flexibility in what's coming and how you use your microscope time at EBIC. BAGs are usually 5 to 15 PIs, and so far we're giving about 230 days every 6 months of cryos time across all 4 of our microscopes. We also have a proprietary or industrial access route, which is right now about 5% of our total time, but that can go up to 10%. Acceptance criteria is always based on scientific excellence, and it's subject to standard Diamond terms and conditions, which is most notably that the work should be published. Also, to get time, we require evidence of suitable cryo-EM samples. Travel and subsistence is covered for UK users, and for international users, they can either apply through iNext or the time on the microscope is free of charge. So some of the software changes that we made in the last year. We have an automatic processing pipeline that kicks in as soon as data starts collecting. And we made improvements to this pipeline. So now there's a startup window that allows you to input certain criteria for better data processing. We've also improved the reliability of the automatic processing pipelines. However, there's still some work to do. Also, last year, unfortunately, we lost our dedicated EM developer for our automated processing pipeline, and we lost a senior staff member that was helping. So we're currently in the process of bringing on new people to further this project. Also, as Tom mentioned, Servos is going to be available soon through CCPEM, and I think ultimately the goal is try to integrate this into our pipeline. So for 2018, we're currently working on a new computer room, which is being wired up. That can be seen in the top picture. And this will allow more GPU time for our users to process their data. We're also hiring new staff members to help with this pipeline implementation. And we're completing integration of our automatic pipeline into iSpy-B. So currently, when our pipeline runs, you can log into iSpy-B and see certain stats about your run, such as defocus for each micrograph. You can see thumbnails. You can get a general idea of data quality, but we hope to further improve this. We're also working on new methods for image compression because we're collecting more and more data, so we're trying to get better at storing these large quantities of data. And then we're also testing new pipelines for data processing, and that's with the help of CCPEM. So this takes me to, I want to talk about a few of our in-house research projects, and that's primarily cryo-focused ion beam milling and cryo-electron diffraction. So for those who don't know, cryo-focused ion beam is a method for preparing thick samples at cryo conditions. So what it allows you to do is take cells that are too thick for imaging and mill them down to 200 or 300 nanometers thick, and it allows you to image your complex of interest in its native state of the cell. This is all done while maintaining the samples at vitreous temperatures of negative 170 about, and it allows us to actually get cellular contacts without the need for resin or dehydration or negative stains. So the process usually involves growing your cells on grids, plunge freezing just like you would do for a normal sample, and then transferring it into the SEM where you can either cut lamella, which is shown on the bottom, or wedges to thin the sample. So on the right is our SIOS at EBIC. It's equipped with a Quorum Sage, and it's been optimized for preparation of frozen hydrated lamella. We hope to upgrade this to a full FEI solution later this year, but currently this method is working well for us, so it's what we have. We initially, so development of our system was actually started with the Kai Grunwald's group, and then was later moved to EBIC at the beginning of 2017. So as an example, I have some yeast as is what we use as a commissioning sample. And so on the top is an SEM image. Is there, yeah. Oh. Anyways, so the top is an SEM image of yeast that have been plunge frozen on an EM grid, which have been milled. The bottom left image is just a low mag TEM image of the same region. And if we zoom in, we can actually collect 3D tilt series and reconstruct into tomograms of these regions. And you can see in the bottom left-hand corner here, you can see mitochondria along with ribosomes and other cellular features. Yeah, thank you. All right. So the next thing I want to talk about is we've also started implementing cryo-electron diffraction. And one of the benefits of electron diffraction is that you have a stronger interaction with matter, which means that you can get strong scattering using home sources or using transmission electron microscopes without the need for synchrotrons. So far, there's been about 27 publications of electron diffraction or microelectron diffraction data. But one of the problems is that it requires thin samples. And so this is currently done by either doing thin regions of the crystals or using some sort of method to break the crystals and make them small enough for imaging. So what we wanted to do is actually use our focused ion beam to thin samples that are too thick for traditional electron diffraction and see if we could solve the structure. So we use lysozyme as a proof of concept. So at the top is an SEM image of a frozen hydrated lysozyme crystal. It measures about 10 microns by 5 high. And here's the SEM image after milling. Here's the same region in the TEM where you can clearly see the crystal along with the buffer that surrounds it. So from that, we were able to collect a diffraction data set in our Talos Arctica and reconstruct it to about 2.5 angstroms. So with that, I just want to thank everyone who helped with this project. So the initial cryo-FIB was worked on with Kai's group, led by Christoph, Christina, Valerie. The yeast was done with Natasha and Helen, and then the lysozyme data was a collaboration between Gwendolf's group, Waju, Dave Stewart, and Felix from FEI. So with that, I want to just make the announcement that we're currently offering our first commissioning call for SIOS time. Initially, we're doing a small group of about 2 to 3 users. Anyone can apply for this time, and you can apply via Diamond's UAS. And if you have any questions, just let me know. And so with that, I'll take any questions.
Hey, sorry. I just have a question about the FIB milling external access. What kind of preliminary data do you need to show for appropriate samples?
Yes, so we'll be sending out an email to anyone on the EBIC, all EBIC users, but what we're requiring for this initial commissioning call is at least low mag images of samples that have been clipped to show that the carbon is mostly intact after clipping, to show that you have good concentration of samples, and to suggest that you have proper vitrification. Even though you won't be able to see that inside of the cells, we are looking for some sort of evidence that there is proper vitrification. And so when we're selecting users, we'll be putting emphasis on the scientific merit of the project, but also the feasibility and its ability to meet sort of those 3 criteria.